Differentiation and functions of CD4 plus effector T cells. So we'll look into overview of CD4 T cells, mediated immune response, subset of CD4 plus T cell, effector cell, T cell, the properties of T helper 1, T helper 2, and T helper 17 subset, development of T helper 1, T helper 2, T helper 17 subset. We look on the T helper 1 subset, development of T helper 1 cells, function of T helper 1 cells. We'll look onto the T helper 2 subset, development of T helper 2 cells, function of the T helper 2 cells. Also the same to 17. We'll look on the function of other helper T cell subset, other subset of CD4 plus effector T cells, and other cytokine producing T cells. So without further ado, let us go on. Defense against microbes that is mediated by T cell is called cell-mediated immunity. T cell provides protection against intracellular and extracellular pathogen and also assist in elimination of tumor cells. Historically, immunologists divided the adaptive immunity into humoral immunity, which is in experiment with animal, can be transferred from immunized donor to naive host by antibodies, and cell-mediated immunity, which can be transferred not by antibody but by T lymphocyte. Humoral immunity neutralize and eliminate extracellular microbes and toxins that are accessible to antibodies. However, antibodies cannot attack microbes that survive inside phagocytes and other cells. T cell mediated immunity evolved to provide defense against such microbes. T cell can also enhance killing of microbes that are normally live and replicate outside cells, but are killed upon ingestion by phagocytes that are activated by T cell derived cytokines. Therefore, defect in cell-mediated immunity results in increased susceptibility to infection by viruses and bacteria that are obligatory intracellular microbes, as well as some of extracellular bacteria and fungi that are eliminated by phagocytes. T cell-mediated reaction are also important in allographic rejection, we see in chapter 17, and tumor immunity, see in chapter 18, and hypersensitivity disease, chapter 19. The two major classes of T cells, these are CD4 plus cell and CD8 plus, function in different and complementary ways in cell-mediated immune reactions. The hallmark of CD4 plus effector T lymphocyte is that they function mainly through secreting cytokines. They serve a critical role in phagocyte-mediated elimination of microbes, which is historically definition of cell-mediated immunity. CD4 plus T cells also activates other leukocytes, including the neutrophils and eosinophil, and stimulate the antibody production by B cells. CD8 plus effector cells kill infected cell and tumor cells and are responsible for the eradication of microbes, typically viruses that survive and replicate inside any cells, including non-phagocytic cells. In this chapter, we will describe the role of CD4 plus T cells in eliminating microbes. At the end, we will discuss some of the less numerous population of T cells whose roles in defense and disease are not well defined. The differentiation and function of CD8 T effector cell will be discussed in chapter 11. Overview of CD4 T cell mediated immune response. The sequence of events in the response of CD4 T cells involve the initial activation of these cells in lymphoid organs to generate effectors and the memory cells. Migration of many of these effector cells to site of infection and elimination of infection pathogen at these sites. We described the early step in activation of T cells in chapter 9, and we will describe the generation and function of effector CD4 plus T cells in this chapter. So normally we have a diagram here, which it will be lovely if you visit the book and see it. We have the A and B. For A is the the one with which generally the diagram is explaining the role of T cell in eradicating infection. A is with CD4 T cell recognizing antigen of phagocytosed and extracellular microbes and produce cytokine and cell surface molecule that recruit and activate the phagocytes to kill microbes. So CD8 T cells can also secrete some cytokines and participate in similar reaction, not shown in that diagram. However, in B, the CD8 cytotoxic T lymphocyte recognize antigen of microbes residing in the cytoplasm of infected cell and kill the cell. So these are two mechanisms which are used in different T cells. However, you see the CD4 is more of a helper cell that helps the macrophage to kill the microbes itself. But when it comes to CD8, it kills the microbes with the cell which owns that particular microbes. So that's the kind of difference. Let us continue. Effect of CD4 plus T cells are generated in secondary lymphoid organs, and most of the effector cells leave these organs and migrate to peripheral site of infection, where they function to eliminate microbes. This migration of effector T cells to site of infection is dependent on endothelial adhesion molecule and chemokine expression at these sites. We have seen already in chapter 3. Although migration is largely independent of antigen, T cells that recognize antigen in the tissue may be preferentially retained there. Once in the tissue, the T cell encounter microbial antigen presented by macrophage and other antigen presenting cells, the antigen presenting cell. T cell that specifically recognize antigen receive signal through their antigen receptor that increases the affinity of integrin for their ligands. Two of these integrins are VLA4 and 5, very late antigen 4 and 5. So those are the ones that you will see. So very late antigen 4 and 5 said bind to fibronectin in extracellular matrices and third adhesion molecule. CD44, which is also highly expressed on activated T cell, bind to hyaluronan. In addition, chemokines receptor expressed in activated T cell bind chemokines that are produced in tissue. As a result of this adhesion chemotactic interaction, antigen-specific effector T cells that encounter the antigen are retained at the extracellular site. The T cells are not specific for the antigen that migrate into a site of inflammation. T cell not specific for antigen that migrate into a site of inflammation may die in the tissue or return to circulation through the lymphatic vessel. Some memory T cells also migrate to peripheral tissue using the same adhesion molecules and chemokines receptor as do effector cells. A fraction of CD4 plus T cells that are activated in secondary lymphoid organ do not exit the organs but migrate into lymphoid follicles within the organs, where they help the B cell to produce high affinity antibodies of the different isotopes. These helper T cells are called the T follicular helper T cells, and they are called the T follicular helper T cells. Their development, properties, and function in humoral immune response are described in chapter 11. In cell-mediated immune response against phagocytes microbes, T cells specifically recognize microbes antigens, but recruited phagocyte and other myeloid cells actually destroy the pathogen. Thus, effector T cells of CD4 plus lineage link specific recognition of microbes with activation of other leukocytes that destroy the microbes. This fundamental concept was first appreciated from studies of cell-mediated immunity to the intracellular bacterium Listeria monocytogenes. It was shown that mice previously infected with low, sublethal dose of Listeria were protected from the challenge with high doses that were lethal in previously infected animal. Protection could be transferred to naive animal with lymphocytes, later shown in T cell lymphocytes, from the infected mice but not with the serum. The fluid fraction of the clotted blood that contain antibodies. These results demonstrated that specific protection against an intracellular bacteria infection was mediated by T cell. However, in vitro, the bacteria were killed not by T cell from the immune animal but by activated macrophage, emphasizing the central role of macrophage in microbial elimination. Thus, such studies established that defense against intracellular microbes required a cooperation interaction between antigen-specific T cells and microbial phagocytes, and we now know this type of interaction is an important component of cell-mediated immunity. Ingression and elimination of microbes by phagocytes is also the major reaction of innate immunity, but T cell greatly enhance this function of phagocyte. As we discussed in chapter 4, phagocytes recognize microbes and are activated by pathogen-associated molecule pattern such as toll-like receptor ligands, and they are capable of destroying variety of microbes. However, many infection pathogens have evolved to revisit this mechanism of innate immunity and survive and even replicate inside macrophage. In such infection, T cells recognize microbial protein, antigen, and activate phagocyte, enabling them to destroy microbes that may not be eliminated by innate function of the macrophage in absence of T helper. CD4 effector T cell activate phagocytes via surface molecule, principally CD40 ligands, and secreted cytokine. We will see how these signals cooperate when we discuss the activation of macrophage later in this chapter. So we have the diagram. As always, I insist you to have a visit on the book and see the diagram, how it looks like. This is figure 10.2. It's the step in CD4 T cell mediated immune response. The CD4 T cells recognize peptide that are derived from protein antigen and presented by dendritic cell in secondary lymphoid organs. The T lymphocytes are stimulated to proliferate and differentiate into effector and the memory cell, which enter the circulation and migrate to the site of infection in peripheral tissue. In the tissue, effector T cells recognize the antigen and respond by secreting cytokine that recruit more leukocytes and activate phagocytes to eradicate the infection. So, if you look closely to the diagram itself, I'll go here slowly with you, and we know for sure this is the tissue site of infection. What is happening here? The dendritic cell is capturing the organism. It has taken it, and it will take back to lymphoid in the lymph node. In the lymph node, for example, lymph node. Then in the lymph node, that antigen recognition of induction in lymphoid organs. So here, naive CD4 T cell will be met, and they will interact. Then this one will be activated. When it is activated as normal, it produces they can proliferate and differentiate, but again they can produce some cytokines and they can cooperate. Then this one will help also the macrophage to kill the organism. So T cell proliferation differentiation, differentiated CD4 T helper cell enter the circulations. Migration of effector T cell and other leukocyte to the site of. So if it inserts any, there will be migration, and some of them, these which have been prepared, the T cell, they will go direct to the place whereby this infection was happening. So this is the site of infection. In here, phagocytosis and killing of microbes is happening because these activated cells, they will connect directly to macrophage, and they are connecting direct to macrophage. The macrophage is able to kill the neutrophil monocyte, and inflammation is happening around. So have time to go around and see what is happening. However, let us continue. Inflammation consisting of leukocyte recruitment and activation accompanies many of the reaction of CD4 plus T lymphocyte. These T cells dependent inflammation serve as an antimicrobial defense mechanism, but also can be injury to tissue. When a T cell reaction causes injury, it is called delayed type hypersensitivity, DTH. The term hypersensitivity refers to an excessive or damaged immune response. The delayed type hypersensitivity frequently occur together with the protective cell-mediated immunity against the microbes and may be cause of much of the pathology associated with certain types of infectious and autoimmune disease, as we will see in chapter 16 and chapter 19. Because the function of CD4 plus T cells are mediated in the large part by cytokines, there have been a great interest in defining these cytokines, which cells produce them, and how they function. An important discovery in immunology was the identification of the population of CD4 plus effector T cells that produce different cytokines and therefore perform distinct function. We will begin with the description of the major properties of these subsets and then describe the development and function of each population. So, subset of CD4 plus effector T cell. There are four major subsets of CD4 plus effector T cells, three of which called Th1, Th2, and Th17 secrete different sets of cytokines, function in host defense against distinct types of infectious pathogen, and are involved in different types of tissue injury in immunological disease. The fourth subset, T follicular helper cells, is important in antibody-mediated immunity and is discussed in chapter 12. Regulatory T cells are another distinct population of CD4 T cells. They are not effector cells. Rather, their function is to control immune reaction to self and foreign antigen, and they are described in chapter 15 in the context of immunological tolerance. So they have the figures here, and I think you can go through, but let us continue. Properties of T helper 1, T helper 2, and T helper 17 subsets. It was appreciated many years ago that host response to different infections vary greatly, as do the reaction in different immunological disease. For instance, the immune reaction to bacteria that survive within phagocytes, like Mycobacterium tuberculosis, is dominated by activated macrophage, whereas the reaction into helminth parasite consists of the production of immunoglobulin E, IgE, antibody, and activation of eosinophil. Furthermore, in many chronic autoimmune disease, tissue damage is caused by inflammation with accumulation of neutrophil and macrophage, whereas in allergic disorder, the lesion contain abundant eosinophil along with other leukocytes. The realization that all of these phenotypical diverse immunological reactions are dependent on CD4 T cell raised an obvious question. How can the same CD4 cells elicit such different response? The answer, as we know now, is that CD4 T cells consist of subset of effector cells that produce distinct set of cytokines, elicit quite different reaction, and are involved in host defense against different microbes as well as in distinct type of immunological disease. The first two subsets that were discovered were called type 1, type 2 helper T cells, that is Th1, Th2. The Th17 subset, so named because it is characteristic cytokine is interleukin 17, was identified many years later as the T cell responsible for some CD4 T cells mediated inflammatory disease that could not be attributed to T helper 1, T helper 2 subset. The defining characteristics of differentiated subset of effector cells are the cytokines they produce, which is related to the transcription factor they express. The transcription factors are responsible for production of different cytokines by this subset, as well as expression of different chemokines, receptors, and other proteins. These characteristics of each subset are described in the following section. The signature of cytokine produced by the major CD4 plus T cell subset are interferons, gamma, for Th1, interleukin 4, interleukin 5, and interleukin 13 for Th2 cells, interleukin 17 and interleukin 22 for Th17 cell. The cytokine produced by these T cells subset determine their effector function and roles in disease. Some of the cytokines made by each subset also stimulate the development and expansion of that subset and inhibit differentiation of other T helper subset, thus contributing to amplification of each types of helper T cell response as process called polarization, discussed later. The production of distinct set of cytokine is initiated by expression of subspecific transcription factor and is sustained by epigenetic modification of specific cytokine gene lock. These mechanisms are described later. T helper 1, T helper 2, T helper 17 cells have distinct pattern of homing, in large part because they express chemokines receptor and adhesion molecule that direct them to migrate in different sites of infection. We discussed the control of lymphocyte migration in chapter 3. T helper 1 but not T helper 2 cell expresses high level of chemokine receptor CXCR3 and CCR5, which bind to chemokine produced in tissue during innate immune response. Therefore, T helper 1 cells tend to be abundant at site of infection where the infection agent triggers strong innate immune reactions. These antigens include many bacteria and viruses. T helper 1 cells also expresses high level of ligand for selectin and P-selectin, which assist in the migration of these cells to site of strong inflammation, where the selectins are expressed on the endothelium. In the contrast, T helper 2 cells expresses the chemokine receptor CCR3, CCR4, and CCR8, which recognize chemokines that are highly expressed at the site of helminthic infection and allergic reaction, particularly in mucosa tissue, and so Th2 cells tend to migrate to this tissue. The Th17 cells expresses CCR6, which bind to the chemokines CCL20, which is produced by various tissue cells and macrophage in some bacteria and fungal infection. Very important for you to remember, and for me as well, that Th1 expresses high level of chemokine receptor CXXR3 and CCR5. But when you come to Th2, it expresses CCR3, no X here, and CCR4 and also CCR8. These are produced by T helper 2. And when you come to 17, it expresses CCR6 and chemokine CCL20, which are produced in various tissue cells and macrophage in bacterial and fungal infection. As you'll see them, these are related that these mostly kill fungus and bacteria. So in summary, these figure 10.4 properties of major subset of CD4 helper T cells. Naive CD4 T cells may differentiate into distinct subsets of effector cell in response to antigen, co-stimulator, and cytokines. Each subset acts mainly on another cell of the immune system referred to the target cell and serves different function and roles in disease. T follicular helper T cells are discussed in chapter 12, interleukin, earliest interleukin. So here you see when you go directly to this diagram, you'll see that this figure, you see T helper 1, this is the effector cell. Defining cytokine, what does it produce? It produces mostly interferon gamma, and principal target cells here, they have macrophage, and major immune reaction is macrophage activation. And what happened in the host defense, it is intracellular, works with intracellular pathogen, and role in disease, it is also involved in autoimmune chronic inflammation. When you come to T helper 2, this is mostly produces interleukin 4, 5, and 13, and principal target cell is eosinophil, and major immune reactions are eosinophil, mast cell activation, alternative macrophage activation. Then host defense is mostly working to prevent or to work with fight with helminth, and it's mostly the role in the disease is mostly allergic. Then we have T helper 17. It produces mostly the interleukin 17 and interleukin 22. These are found in neutrophil mostly, and neutrophil recruitment is the main major immune reaction, and it works for extracellular bacteria and fungus. Also, it is involved in autoimmune and inflammation. And you'll find in autoimmune and inflammation, the T helper 2 and T helper 17 are more common. Then we have T follicular, T helper 2 follicular, TH2, the CD4 T follicular. This produces the most common are interleukin 21. This is and interferon gamma together with interleukin 2, interleukin 4. They work mostly in B cell, and the main function is for antibody production. They work with the cellular pathogen when happen and the autoimmune antibodies. So that is a summary of what is happening with a CD4 T cell. Although for many years it was thought that T helper 1 and T helper 2 cells help B lymphocyte to produce different antibodies, it is now clear that, as stated earlier, most of these differentiated effector cells leave the lymphoid organ where they are generated and migrate to peripheral sites of infection. Antibody response develop mostly in secondary lymphoid organs and particularly in the germinal center where the antigen specific B and T cell interact. The CD4 plus helper T cells that remain in secondary lymphoid organ to help B lymphocyte are not classical T helper 1, T helper 2, but T follicular helper cells that make some of the same cytokine that interleukin the T helper 1 and T helper 2 are making. See in chapter 12. Different inflammatory diseases are caused by excessive reaction of different helper T cell subset. In general, T helper 1 and T helper 17 cells play a prominent roles in autoimmune diseases associated with inflammation, whereas the allergic reaction are dominated by T helper 2 cells. The identification of T helper 1, T helper 2, T helper 17 subset have provided many insight into the lymphocyte response. However, there are some covent with the idea that all effector CD4 T cells can be classified into these subsets based on a defined criteria. Many CD4 T cells, effector T cells, produce combination of cytokines or only some of the cytokine characteristics of particular subset and are not readily classifiable into separable population. For instance, in many inflammation reactions, there may be an individual T cell that produces both interferon gamma, characteristic of Th1 cell, and interferon and interleukin 17, typical of Th17 cells. Conversely, some cells may produce cytokines that are not characteristics of any of three subsets, such as interleukin 9 or only some of the cytokine produced by a particular subset. These restricted cytokine profile have led to an expanded nomenclature describe these populations such as T helper 9, T helper 22, and so on. It is not known whether cells with mixed or limited cytokine pattern are intermediate in the development of the classical polarized effector cells, or are themselves fixed population. It is also clear that some of these effector T cells may convert from one cytokine profile to another in response to changes in activation condition. For the extent and significance of plasticity or stability of differentiated effector T cell remain topic of active research. Although CD4, wait a little bit. Although CD4 effector T cells and are the main source of many cytokines in protective and pathological adaptive immune response, the same cytokine may be produced by other cell types such as gamma delta T cells and innate lymphoid cells, as we discussed in chapter 4. Innate lymphoid cells are classified into the group that produce many of the same cytokine as subset of effector CD4 T cells do. For this reason, the concept has evolved that cytokine-dependent host defense and pathological reaction are mediated by a coordinated action of innate lymphoid cells in response to effector CD4 T cells later. Such response may be viewed as type of immunity. Type 1 immunity relies on innate lymphoid cytokine 1, natural killer cells, and T helper 1 cells. Type 2 immunity on innate lymphoid cells and T helper 2 cells, and type 3 immunity on innate lymphocyte 3 and the T helper 17 cells. Development of T helper 1, T helper 2, and T helper 17 subset. Differentiated T helper 1, T helper 2, T helper 17 cells all develop from naive CD4 T cells lymphocytes, mainly in response to the cytokine present early during immune response. The process of the effector T cell development involves multiple steps. Signal that T cell receive from antigen-presenting cell and other cells at the site of immune response initiate the conversion of antigen-stimulated T cell to effector cells. Development of effector cell become progressively committed to a particular cytokine production profile, and cytokine amplify this differentiation pathway. The net result is the progressive accumulation of T cell population that produce a distinct set of cytokines. There are several important general features of T cell subset differentiation. The cytokine that drive the development of CD4 T cell subset are produced by antigen-presenting cell, primarily dendritic cells and macrophage, and other immune cells such as natural killer cells and mast cell present in the lymphoid organ where the immune response is initiated. Dendritic cells that encounter microbes and display microbial antigen are activated to produce cytokine as well as co-stimulators as part of innate immune response to microbes. Different microbes may stimulate dendritic cell to produce distinct set of cytokine, perhaps because microbes activate different dendritic cell population are recognized by different microbial sensor in cell. Other cells of innate immunity, such as natural killer cells, innate lymphoid cells, and mast cell, also produce cytokine that promote development of antigen-stimulated T cell into different effector subsets. Stimuli other than cytokine influence the pattern of helper T cell differentiation. Experimental evidence indicate that the affinity of the T cell receptor for antigen, the amount of antigen, and the nature of antigen presenting cell determine the dominant T subsets that develop after antigen recognition. But the contribution for this factor to development of effector subset is in most immune response is not clear. The genetic makeup of the host is another important determinant of the pattern of T cell differentiation. Some inbred strain of the mice develop T helper 2 response to the same microbe that stimulate Th1 differentiation in most other strain. Strain of the mice that develop Th2 dominant response are susceptible to infection by intracellular microbes. It is important not to prove that people differ in their propensity to mount T helper 1, T helper 2, or T helper 17 response based on inherited genes. The distinct cytokines profile of differentiated cell population are controlled by particular transcriptional factor that activate cytokine gene expression and by chromatin modification affecting accessibility of these factor to the promoters and regulatory element of cytokine genes. The transcriptional factor are activated or induced by signal from T cell receptor, innate immune receptor, co-stimulators, and cytokine receptor. Each of subset expresses its own characteristic set of transcriptional factor. As the subset become increasingly polarized and gene lock encoding that subset signature, cytokine undergo histone modification, such as change in methylation and acetylation, the other chromatin remodeling events, so that these lock remain accessible to RNA polarize the transcription factor, whereas the lock of other cytokine, those not produced by that subset, are inaccessible to chromatin state. Thus, the cytokine genes characteristics of particular subset become fixed in the antigen response state, whereas the gene that encode cytokines not produced by that subset remain inactive. These epigenetic changes are inherited in progenitors of the proliferating cells, thus ensuring that the effector T cell become stably committed to producing a particular set of cytokines. Each subset of differentiated effector cell produces cytokine that promote its own development and may suppress the development of other. These features of T cell subset development provide a powerful amplification mechanism. For instance, interferon gamma secreted by T helper 1 cell stimulate the further T helper 1 differentiation and inhibit the generation of T helper 2 and in T helper 17. Similarly, interleukin 4 produced by T helper 2 cells promote T helper 2 differentiation. Thus, once immune response develop along one effector pathway, it becomes increasingly polarized in that direction, and most extremely polarization is seen in chronic infection and or upon chronic exposure to environmental antigen when immune stimulation is prolonged. Differentiation of each subset is induced by types of microbes that the subset is the best able to combat. For instance, the development of T helper 1 cells is driven by the intracellular microbes against which principal defense is T helper 1 mediated. By contrast, the immune system responds to Helminth's parasite by development of T helper 2 cell, and cytokines produced by these cells are important for combating Helminth. Similarly, T helper 17 response are induced by some bacteria and fungi and most effective at defending against these pathogens. And the generation and the effector function of these differentiated T cells are excellent illustration of the concept of specialization of adaptive immunity, which refer to the ability of the immune system to respond to different microbes in a way that are optimal for combating those microbes. With this background, we proceed to a description of the development and function of each subset. So this is the development of T helper 1. Let us go on. The Th1 subset. The interferon gamma producing Th1 subset is induced by microbes that are ingested by and have evolved to survive and replicate within a phagocyte. It is the major effector T cell population in the phagocyte mediated host defense. Th1 cells were the first defined subset of helper T cells shown to mediate cellular immunity against pathogens that survive inside the phagocytes. So this is the cell development. You see that dendritic cells, it has the antigen, and these are microbes. So they have been involved. They have been ingested. Then in naive T cell, it is made in contact with it. This causes the production of interleukin 12. This interleukin 12 is going there to activate these naive cells and other cells to survive, and this one is changed to activated cell. So we have these cells now with Th1 characteristics with Tbet, STAT1, STAT4, and T cell receptors amplification. They are producing interferons. The interferon is taken again, and again is keeping proliferating cells. So we say development of Th1 cells, interleukin 12 produced by dendritic cells and macrophage in response to microbes, including intracellular microbes, and interferon gamma produced by natural killer cells. All part of this early innate immune response to the microbes activates transcription factors Tbet, STAT1, and STAT4, which stimulate differentiation of naive CD4 T cells to T helper 1 subset. Interferon gamma produced by T helper 1 cell amplify this response and inhibit the development of T helper 2 and T helper 17. Other cytokines including interferon, type 1 interferon, and interleukin 18 also promote T helper 1 differentiation but not shown. So dendritic cell, they produce this, and also the natural killer cell is responsible for the production of interferon. So all interferon together with the interleukin 12, they can change the naive T cell to become T helper 1. The T helper 1 cells, this is the one, by activating those the transcription factors. Development of T helper 1 cell. T helper 1 differentiation is driven by mainly cytokine 12 and interferon gamma, and occur in response to microbes that activate dendritic cell, macrophage, and natural killer cell. The differentiation of antigen activated CD4 T cells to T helper 1 effector is stimulated by many intracellular bacteria such as Listeria and Mycobacteria, and by some parasites such as Leishmania, all of which infect dendritic cell and macrophage. T helper 1 differentiation is also stimulated by viruses and by protein antigen administered with the strong adjuvant. A common feature of this infection and immunization condition is that they elicit the innate immune reaction that are associated with the production of certain cytokines, including interleukin 12, interleukin 18, and type 1 interferons. All of these cytokines promote T helper 1 development of these. Interleukin 12 is the probably the most important. Many microbes stimulate natural killer cell to produce interferon gamma, which itself a strong Th1 induced cytokine, and also acts in dendritic cells and macrophage to induce more interleukin 12 secretion. After TLP1 cells have developed, they secrete interferon gamma, which promote more TLP1 differentiation and thus amplify the reaction. In addition, interferon gamma inhibits the differentiation of naive T cells to T helper 2, T helper 17 subset, thus promoting the polarization of immune response in one direction. T cell may further enhance cytokine production by dendritic cell and macrophage by virtue CD40 ligand on activated T cells engaging to CD40 on antigen presenting, stimulating the interleukin 12 secretion. Interleukin gamma and interferon gamma and interleukin 12 stimulate the Th1 differentiation by inducing and activating the transcription factor TBET, STAT1 and STAT4. TBET, a member of T-box family of transcription factor, is induced in naive CD4 T cells in response to antigen and interferon gamma. Interferon gamma also activates STAT1, signal transducer and activation transcription 1, which is in turn stimulate expression of TBET. TBET then promote interferon gamma production through a combination of direct transcription activation of IFNG genes and by inducing chromatin remodeling of IFNG promoter region. The ability of interferon gamma to stimulate TBET expression and the ability of TBET to enhance interferon gamma transcription set up a positive amplification loop that drive differentiation of T cell toward TLP1 phenotype. Interleukin 12 contribute to the Th1 commitment by binding to receptors on antigen stimulated CD4 T cells and activating the transcription factor STAT4, which further enhance interferon gamma production. Function of T helper 1 cell. The principal function of T helper 1 cell is to activate macrophage to ingest and destroy microbes. The same reaction of T helper 1 mediated macrophage activation is involved in injuries of delayed T cell hypersensitivity, which are component of many inflammatory diseases. Some of these conditions are characterized by granulomatous inflammation, including infectious diseases such as tuberculosis and chronic inflammatory disease such as sarcoidosis. Before discussing the activation of macrophage and how they destroy microbes, we will describe the properties of interferon gamma, the cytokine responsible for the most specialized function of T helper 1 cell. Interferon gamma. Interferon gamma is the principal macrophage activating cytokines. Interferon gamma is also called immune or type 2 interferons. Although in its name interferon is shared with antiviral type 1 interferons, is not a potent antiviral cytokines and its function mainly as activator of effector cell of immune system. Interferon gamma is an homodimeric protein belonging to type 2 cytokines family, seen in chapter 7. In addition, CD4 plus T helper 1s and innate lymphocyte cells, natural killer cell and CD8 T cells also produce interferons. Natural killer cells secrete interferons in response to activating ligands on surface of infected or stressed host cell, or in response to interleukin 12. In this setting, interferon gamma function as mediator of innate immunity. In adaptive immunity, T cell produce interferon gamma in response to its antigen recognition and production is enhanced by interleukin 12 and interleukin 18. So this is the classical macrophage activation, enhance the microbial killing. As we see, function of T helper 1 cells. T helper 1 cells secrete interferon gamma, which act on macrophage to increase the phagocytosis and killing of microbes in phagosome. T helper 1 also produce tumor necrosis factor, which activates neutrophil to promote inflammation. Not shown here, but this is what is happening. So we have antigen presenting cell, the dendritic cell assumed with the bacteria inside, then it activates the naive T cell. The naive cell T cell, which is activated, produces interferon. The interferon activate this one to undergo the process of killing. So the receptor of interferon gamma is composed of two structurally homologous properties. Polypeptide belong to a type 2 cytokine receptor family called interferon gamma R1 and interferon gamma R2. Interferon gamma bind to and induces the dimerization of two receptor chain. This leads to the activation of associated Janus kinase JAK1 and JAK2, and ultimately to phosphorylation and dimerization of STAT1, which stimulate the transcription of several genes, seen in Chapter 7. Interferon gamma-induced gene encodes many different molecules that mediate the biological activities of this cytokine described next. Interferon gamma activates macrophage to kill phagocytes microbes. Interferon gamma works with the other signal to induce the type of macrophage activation that is called classical activation, discussed more in detail later. Interferon gamma promotes the differentiation of CD4 T cells to T helper 1 subset and inhibit the development of T helper 2 and T helper 17. These effects of interferon gamma because it induces the expression of transcription factor needed for T helper 1 development and inhibit transcription of factor involved in T helper 2, T helper 17 differentiation. Interferon gamma stimulates expression of several different proteins that contribute to the enhanced antigen presentation and T cell activation. The proteins include major histocompatibility complex molecule, many proteins involved in antigen processing, including component of proteasome and B7 co-stimulators on antigen-presenting cells. Interferon gamma activate macrophage, dendritic cell, and other cells to produce cytokines that amplify the host response. These cytokines include tumor necrosis factor, interleukin-1, and chemokine, which recruit more leukocyte and induce inflammation. Interleukin-12, which provide a positive feedback loop for interferon gamma production and T helper 1 development. Other T helper 1 cytokines. In addition to interferon gamma, T helper 1 cells produce tumor necrosis factor, which contribute to the recruitment of leukocyte and enhances inflammation. Somewhat surprisingly, T helper 1 cells are also a source of interleukin-10, which function mainly to inhibit dendritic cell and macrophage, and thus to suppress the T helper 1 activation. This is an example of negative feedback loop in T cell response. T helper 1 mediated classical macrophage activation and killing of phagocyte microbes. T helper 1 cell activate macrophage through a contact-mediated signal derived by CD4 ligand CD40 interaction and by interferons gamma. This pathway of macrophage activation is called the classical macrophage activation, to distinguish it from the alternative macrophage activation induced by T helper 2 cytokine described later. Classical activated macrophage are also called M1 macrophage. When T helper 1 cells are stimulated by antigen, the cell expresses CD4 ligands on the surface and secrete interferon gamma. The action of interferon gamma on macrophage described earlier synergize with the action of CD40 ligand, and together they potent stimuli of macrophage activation. The CD40 signal activates the transcription factor NF kappa beta, nuclear factor kappa beta, and up activation protein 1, and as discussed earlier, the AFN gamma interferon gamma activate the transcription factor STAT. This transcription factor together induces the expression of gene encoding several enzymes that localize in phagosomes of macrophage, including inducible nitric oxide synthetase, which stimulate the production of nitric oxide, and lysosome enzyme. Macrophage activation is also associated with the assembly of enzyme phagocyte oxidative in membrane of phagocytes, which induce the production of reactive oxygen species, although this is less prominent than neutrophil. The recruitment of interaction between the surface molecule CD40 and the macrophage and CD40 ligand on T cell ensures the macrophage that they are presenting presenting antigen to T cells. This is macrophage that are harboring intracellular microbes, are also the macrophage that will be in contact with T cells and thus most efficiently activated by T cells. Activated macrophage kills phagocytes microbes mainly by the action of nitric oxide and reactive oxygen species and lysosomal enzymes. All of these potent microbial agents are produced within lysosome of macrophage and kill ingested microbes after phagosomes fuse with the lysosomes. These toxic substances also may be released into adjacent tissues where they kill extracellular microbes and may damage normal tissue. Inherited immunodeficiencies have been established. Inherited immunodeficiencies have established the critical importance of T helper 1 cells in cell-mediated immune against intracellular pathogen. Different homozygous mutation affecting the interferon gamma receptor, interleukin 12 and interleukin 12 receptor, and STAT1 causes defect in development of T helper 1 cells. Patients with this inherited mutation are susceptible to infection with the Mycobacterium tuberculosis, low virulence environmental Mycobacterium, and attenuated Mycobacterium bovis strain used in Bacillus calmette-Guerin, the BCG vaccine. As group, these disorders are called Mendelian susceptibility of Mycobacterium disease. The patient are also susceptible to infection with other intracellular bacteria such as Salmonella and Protozoa parasite, further establishing the critical role of T helper 1's response in defense against intracellular microbes. Rare patients make autoantibody against their own interferon gamma and are also susceptible to the Mycobacterium infection. Human with inherited mutation in CD40 ligands, X-linked hyper IgM syndrome, and mice which the genes of CD40 ligands and CD40 and CD40 ligand is knocked out are highly susceptible to infection with intracellular microbes, notably the fungus Pneumocystis jirovecii, which require T-cell dependent macrophage activation to be eradicated. These patient and knockout mice also have defect in helper T-cell dependent antibody production because of the critical role of CD40 ligands, CD40 interaction in B-cell activation. We'll see it in chapter 12. So we have another diagram here, figure 10.7. Let us read, then see the diagram. Macrophage activation by T helper 1 cell A. We have the diagram A and B. Macrophage are activated by CD40 ligands, CD40 interaction, and interferons gamma expressed by T helper 1 cells and perform several functions that kill microbes, stimulate inflammation, and enhances antigen-presenting capacity of the cell. So here we see in one the activation of macrophage. So this is macrophage. What is happening? Macrophage ingested bacteria, and there is interaction of ligands from the T cell and CD40 from the macrophage. Then this one leads the interferon gamma to be produced by T cell, the CD4 effector T cell T helper 1. Then if this is produced, you know the function of this one. It will lead to the production of a lot of the enhanced cell will produce reactive oxygen species together with nitrogen oxide. All of these together with lysosome, they will kill bacteria which is inside. So this is the main response that is occurring when these things are happening. They interact together. So B, the principal response of macrophage activated by a classical activation pathway and their roles in T-cell mediated host defense are listed. Macrophages are also activated during innate immune reaction and perform similar function. You see. So in B here we see macrophage response, production of reactive oxygen species, nitric oxide, increasing lysosomal enzymes. The roles in cell mediated, this is involved for the killing of microbes in phagocytosome, effector function of macrophage. Then there is secretion of cytokines like tumor necrosis factor, interleukin 1, interleukin 12, and chemokines. This one, the tumor necrosis factor, interleukin 1, chemokines, leukocyte recruitment, especially for inflammation. The interleukin 12 T helper is responsible for T helper 1 differentiation and the interferon gamma production. Increase the expression of B7 co-stimulator. This is important. Increase T cell activation and amplification of a T cell response. So that's what was happening. Let us continue. Macrophage activated by T helper 1 cells are involved in several other reactions of the host defense. You see figure 10.7. And we have seen it already. They stimulate inflammation mainly through the secretion of cytokines, including tumor necrosis factor, interleukin-1, which recruit more leukocyte, enhances the host's ability to destroy infectious pathogens. Activated macrophage may amplify cell-mediated immune response by becoming more efficient antigen-presenting cell because of increased level of a molecule involved in antigen processing and increased the surface expression of class 2 molecules and co-stimulators, and by producing cytokines such as interleukin 12 that stimulate the T lymphocyte differentiation into effector cell. Some tissue injury may normally accompany T helper 1 cell mediated immune reaction to microbes because the microbicidal products released by activated macrophage and neutrophil are capable of injuring normal tissue and do not discriminate between microbes and host tissue. These tissue injury usually resolved as the infection is cleared. However, excessive T helper 1 reaction are the cause of the tissue injury in many chronic inflammation disease. We'll see it in chapter 19. Now, T helper 2 subsets. T helper 2 cells activate the defense mechanism that uses IgE antibodies, eosinophil, and mast cell to combat microbes. These reactions are important for the eradication of helminth infections and perhaps also to elimination of other microbes in mucosal tissue. They are central to the development of allergic disease. You'll see in chapter 20. The T helper 2 cells are also thought to be important in tissue repair. Development of T helper 2 cells. T helper 2 differentiation occur in response to helminth and allergic and is dependent on cytokine 4. Because the major cytokine that promote T helper 2 development is interleukin 4, and this is the product of T helper 2 cell. There have been some uncertainty about source of interleukin 4 to initiate the T helper response. The cytokine may be made by antigen stimulating T cells, by mast cell, and possibly by group 2 innate lymphoid cells, innate lymphoid cell 2, see in chapter 4, and other cell in a vicinity of activated T cells. Other cytokines that may promote the development of T helper 2 cells include interleukin 25, interleukin 33, and thalamic stromal lymphopoietin TSLP, all of which are produced by damaged epithelial and other cells and are involved in the activation of innate lymphoid cells too. Interleukin 4 stimulates T helper 2 development by activating a transcription factor STAT6, which together with a T cell receptor signal induces expression of GATA3. The GATA3 is the transcription factor that is required for expression of T helper 2 cytokine interleukin 4, 5, and 13. GATA3 works by directly interacting with the promoters of these cytokines genes and also by causing chromatin remodeling, which open up the locus for accessibility to other transcription factor. This is similar to the way in which the T-bet influences the interferon gamma expression. GATA3 function to stably commit differentiated cell toward the T helper 2 phenotype, enhancing its own expression through the positive feedback loop. Furthermore, GATA3 blocks T helper 2 differentiation by inhibiting expression of signaling chain of the interleukin 12 receptor. Knockout mice lacking interleukin 4, STAT6, and GATA3 are deficient in T helper 2 response. So that's the important concept. The function of T helper 2 cells. The T helper 2 cells stimulate the IgE mast cell and an eosinophil mediated reaction that serve to eradicate helminthic infection and promote tissue repair. Helminths are too large to be phagocytosed by neutrophil and macrophage and may be more resistant to the microcidal activities of these phagocytes than the most bacteria and viruses. Therefore, other mechanisms are needed for defense against helminth infection. The function of T helper 2 cells are mediated by interleukin 5, which activate the eosinophils, interleukin 13, which have diverse actions, T follicular helper cells that produce interleukin 4 and interleukin 13 stimulate the production of IgE antibodies, which are involved in the most Th2 mediated defense reaction. We will first describe the properties of these cytokines and then their roles in host defense. We'll see this one, the development of T helper 2. Let us see first. So, development of T helper 2 cells. The Dendritic cell may respond to cytokine produced in epithelial by becoming T helper 2 inducers by mechanisms that are not well defined. Interleukin 4 produced by activated T cells themselves or the master cell and eosinophil, especially in response to helminth, activate the transcriptional factor GATA3 and STAT6, which stimulate the differentiation of naive CD4 T cell to T helper 2 subset. Interleukin 4 produced by the T helper 2 cells amplify this response and inhibit the development of T helper 1 and T helper 17. The diagram, the way it is in the epithelium when you have the helminth or protein antigens which causes allergy. Then this one, when they're in epithelial, they produce a lot of interleukin 25, interleukin 34, TLSP, of which they are called the inflammasomes together. These they activate the dendritic cell. The dendritic cell also present to naive T cell. In the process, this one is activated. When it is activated, it produces interleukin 4. The T cell naive T cell, if it is interacted with helminth, they produce interleukin 4. Interleukin 4 goes around by itself. Then this one causes more activation of cell, and when interleukin 4 goes within the cell, it causes the activation of STAT6 and GATA3. GATA3 is the one that is responsible to go to the nuclear DNA and lead to the amplification and more further production of interleukin 4. Further production of interleukin 4 is going like a automatic process of doing itself. When T helper 2 cells are produced, they release T helper interleukin 4, interleukin 5, interleukin 13, as we'll see the function later. So interleukin 4. Interleukin 4 is the signature of cytokine of the T helper 2 subset and function as both as inducer and effector cytokine of these cells. It's the member of the type 1, 4 or alpha helicocytokine family. The principal cellular source of interleukin 4 are CD4 T cell lymphocytes of the T helper 2 subset, and also innate lymphoid cells too, and activated mast cell. Interleukin 4 receptors consist of a cytokine binding alpha chain that is the member of type 1 cytokines receptor family associated with the gamma C chain shared by other cytokine receptor. This interleukin 4 receptor gamma YC receptor signal by JAK-STAT pathway involving JAK1, JAK4, JAK3, JAK6 by pathway involve the insulin response substrate protein called IRS2. Activated STAT induces transcription of genes that account for many of the action of these cytokines. Interleukin 4 and interleukin 13 receptor share one of their two chain described later. So this is figure 10.9, the function of T helper 2 cell. The CD4 cell T cell that differentiate into T helper cells secrete interleukin 4, 5, and 13. Interleukin 4 and interleukin 13 act on B cell to stimulate production of antibodies that bind to the mast cell and eosinophil, such as IgE. Help for antibody production may be activated by T follicular helper cells that produce T helper 2. So here we have the helminth. It has present the naive T cell proliferate. Then we have the production of interleukin 4 and 13. These are they activate macrophage, the macrophage, alternative macrophage activation tissue repair. So this is where we'll see the macrophage 2 instead of 1 that we saw there. And also interleukin 4, 13 can go and activate B cell to produce antibodies which are IgE, most of them. They act in mast cell degranulations, and you see the information in the other function. Also, there will be production of IG4 and IG1, IG4 in human and IG1 in mouse. So interleukin 4 and interleukin 13 also can go to intestine, and this intestinal mucosa can lead to the intestinal mucosa production of mucus. When mucus are produced, this helps also to help the parasite to move outside the body. So there are many other functions. The interleukin 5 again can activate eosinophil. When eosinophil goes, this eosinophil activation, it can also produce a lot of toxin to helminth. So this is the fight against the helminth in different pathway: antibody production, um, mucus productions, and many other functions, because the parasite is not easily to be ingested and be killed. Therefore, there are many other mechanisms that should be used. Let us go. So we continue here with this diagram. The cytokine reside lymphocyte organs and not by classical Th2 cell. Interleukin 5 activate the eosinophil, a response that is important for defense against helminth infection. Interleukin 4 and interleukin 13 are involved in immunity on mucosal barrier, induce an alternative pathway of macrophage activation, and inhibit classical 1 mediated macrophage activation. So that's what is happening. So interleukin 4 has important action on several cell types. Interleukin 4 and interleukin 13 produced by T follicular cells stimulate B cell Ig heavy chain class switching to IgE. IgE heavy chain to class 2 to the IgE isotype. The mechanism of class switching are described in chapter 12. IgE is a principal mediator of immediate hypersensitivity, allergic reactions. Interleukin 4 also enhances switching to Ig4 in human and homologous IgG1 in mice, but the significance of this role of interleukin 4 is unclear because these Ig subclasses do not bind to phagocyte Fc receptor or activate the complement. Interleukin 4 stimulates the development of T helper 2 effector cells from naive CD4 T cells and may be a growth factor for differentiated T helper cells. This function of interleukin 4 was described earlier. Interleukin 4 together with interleukin 13 contribute to an alternative form of macrophage activation that distinguishes from macrophage response to interferon gamma. Interleukin 4 and interleukin 13 suppress interferon gamma mediated classical macrophage activation and thus inhibit defense against intracellular microbes that are destroyed by phagocytosis. The alternative macrophage activation pathway is described later. Interleukin 4 and interleukin 13 stimulate peristalsis in the gastrointestinal tract, and the interleukin 13 increases the mucus secretion from airway and gut epithelial cells. Both actions contribute to the elimination of microbes at epithelial surface. Interleukin 4 and interleukin 13 stimulate the recruitment of leukocyte, notably the eosinophil, by promoting the expression of adhesion molecule endothelial and secretion of chemokines that bind chemokines receptor expression on eosinophil. Interleukin 13 is structurally and functionally similar to interleukin 4 and also play a role in defense against helminth, seen in chapter 16, and in allergic disease chapter 20. Interleukin 13 is a member of type 1 4 alpha helicocytokine family. Interleukin 13 is produced mainly by T helper 2 subset, but group 2 innate lymphocyte cells and other leukocytes may also produce cytokines. Interleukin 13 receptor is heterodimer of interleukin 4 receptor alpha chain and the interleukin 13 alpha chain. This complex can bind both interleukin 4 and interleukin 13 with high affinity and also signal through a JAK1, JAK3, JAK1, JAK3 and STAT6 pathway. The receptor is expressed on a wide variety of cells, including B cells, mononuclear phagocytes, dendritic cell, eosinophil, basophil, fibroblast, endothelial cells, bronchial epithelial cells. Interleukin 13 works together with interleukin 4 in defense against helminth and in allergic inflammation. Some of the action of interleukin 13 overlap those of interleukin 4 and other are distinct. Both interleukin 13 and interleukin 4 can activate B cell to switch to IgE and some IgG isotopes and recruit leukocytes, and both are involved in alternative macrophage activation. Interleukin 13 stimulates mucus production by airway epithelial cells, an important component of allergic reactions such as asthma. Unlike interleukin 4, interleukin 13 is not involved in T helper 2 differentiation. Interleukin 5. Interleukin 5 is an activator of eosinophils and serves as principal link between T cell activation and eosinophil inflammation. It is homodimer of a polypeptide containing a 4 alpha helical domain and is a member of type 1 cytokine family. It is produced mainly by T helper 2 cells and innate lymphocyte cells. The interleukin 5 receptor is heterodimer composed of a unique chain and a common beta chains, which is also part of interleukin 3 and granulocyte macrophage clone stimulating factors receptors. The major interleukin 5 induced signaling pathway involve JAK2, and STAT3. The principal action of interleukin 5 are to activate mature eosinophil and stimulate the growth and differentiation of eosinophil. Activated eosinophil are able to kill helminth. Eosinophil expresses Fc receptor specifically for IgA and some IgG antibodies and thereby able to bind microbes such as helminth that are coated with these antibodies. Role of Th2 cells in host defense. Th2 cells function in defense against helminth and other infections by several mechanisms. IgE and eosinophil mediated reaction, interleukin 4 and interleukin 13 stimulate the production of helminth-specific IgE antibodies which may activate mast cell and the site of infection. Interleukin 5 activate eosinophils and these cells release their granule content including major basic proteins and major cationic proteins which are capable of destroying even tough integument of helminth. See in chapter 16. In allergic reaction, IgE coats mast cell and induce their degranulation upon encounter with antigen. So, host defense at mucosal barrier. Cytokine produced by Th2 cells are involved in blocking entry and promoting expansion of the microbes from the mucosal organ by stimulating mucus production and in intestinal peristalsis. Thus, T helper 2 cells play an important role in host defense at barrier with external environment, sometimes called barrier immunity. Alternative macrophage activation and tissue repair. Interleukin 4 and interleukin 13 activate macrophage to express enzymes that promote collagen synthesis and fibrosis. The macrophage response to T helper 2 cytokine has been called alternative macrophage activation to distinguish it from the activation induced by interferon gamma, which was characterized first, hence designation classical, and which results the potent microcidal function and inflammation. Alternative activated, also called M2 macrophage, produce cytokine that terminate inflammation and initiate repair of diverse types of tissue injury. The macrophage as well as T helper 2 themselves induce a scar formation by secreting growth factor that stimulate fibroblast proliferation, platelet-derived growth factor, collagen synthesis, interleukin 13 transforming growth factor beta, and a new blood vessel formation, angiogenesis, fibroblastic growth factor. T helper 2 cytokines also suppress classical macrophage activation and interfere with protective T helper 1 mediated immune response to intercellular infection. Although the separation of classical and alternative macrophage activation provide a useful text for understanding macrophage heterogeneity, numerous other subpopulations have been described, and M1 and M2 macrophage are likely not fixed subset. So 1 minute. The the T helper 17 subset. The T helper 17 subset is primarily involved in recruiting neutrophil and, to a lesser extent, monocyte sites of infection and inflammation. Some cytokine made by T helper 17 cell serves to maintain the integrity of epithelial barrier. These actions are critical for destroying bacteria and fungal microbes that are killed by phagocytes and repairing injured epithelial layer, but they also contribute significantly to the chronic inflammatory disease. Development of T helper 17 cells. The development of T helper 17 cell is stimulated by pro-inflammatory cytokine produced in response to bacteria and fungi. Various bacteria and fungi act on dendritic cell and macrophage and stimulate the production of cytokines, including interleukin 6, interleukin 1, interleukin 23, all of which promote differentiation of CD4 T cells to T helper 17 subset. Engagement of lectin-dectin 1 on dendritic cell by fungal glycan is signal for production of these cytokines. The combination of cytokines that drive T helper 17 cell development may be produced not only in response to particular microbes such as fungi, but also when cells infected with various bacteria and fungi undergo apoptosis and are ingested by the dendritic cell. Whereas interleukin 6 and interleukin 1 stimulate the early steps in the T helper 17 differentiation. Interleukin 22 may be more important for proliferation and maintenance of differentiated T helper 17 cells. A surprising aspect of T helper 17 differentiation is that tumor growth factor beta, which is produced by many cell types and in an anti-inflammatory cytokines, seen in chapter 15, promote the development of pro-inflammatory T helper 17 cell when other mediators of inflammation such as interleukin 6 and interleukin 1 are present. The T helper 17 differentiation is inhibited by interferon gamma and interleukin 4, thereby strong Th1 and Th2 response tend to suppress T helper 17 development. So see the classical figure 10, a classical and alternative macrophage activation. This was the for other one. Please, we are talking of macrophage here, though we are talking of T helper 17. Let us a little bit pass through this diagram. So we have a classical and alternative macrophage activation. Different stimulus activate tissue macrophages to develop into functional distinct population. Classical activated macrophages are induced by microbial product and cytokine, particularly interferon gamma, and are microbicidal and involved in potential harmful inflammation. Alternative activated macrophages are induced by interleukin 4 and interleukin 13 produced by T helper 2 cells and other leukocytes and function to control inflammation and promote tissue repair and fibrosis. Some evidence suggests that M2 macrophage comprise of population of some of which are mainly anti-inflammatory and other responsible for tissue repair. Now is nitric oxide, reactive oxygen species, and other things that we will be talking. However, let us continue with development of T helper 17 cells. I'm repeating again. The development of T helper 17 cell is stimulated by pro-inflammatory cytokine produced in response to bacteria and fungus. Various bacteria and fungi act on the dendritic cell and macrophage and stimulate the production of cytokine including interleukin 6, interleukin 1, interleukin 23, all of which promote differentiation of CD4 T cells to the T helper cell 17 subset. Engagement of lectin-dectin 1 on dendritic cell by fungal glucan is a signal for the production of these cytokines. The combination of cytokines that drive T helper 17 cell development may be produced not only in response to particular microbes such as fungi, but also when cells infected with various bacteria and fungi undergo apoptosis and are ingested by the dendritic cell. Whereas interleukin 6 and interleukin 1 stimulate the early step in T helper 17 differentiation, interleukin 23 may be more important for proliferation and maintenance of differentiated T helper 17 cell. A surprising aspect of T helper 17 differentiation is that tumor growth factor beta, a transforming growth factor beta, which is produced by many cells and it is an anti-inflammatory cytokines, Cs, promote development of pro-inflammatory T helper 17 cells when other mediator of inflammation such as interleukin 6, interleukin 7, interleukin 1 are present. Interleukin 17 differentiation is inhibited by interferon gamma, interleukin 4. Therefore, strong Th1 and Th2 response tend to suppress T helper 17 development. The development of Th2 cells is dependent on the transcription factor ROR gamma T and STAT3. Tumor growth transforming growth factor beta and inflammatory cytokine, mainly interleukin 6 and interleukin 1, work cooperatively to induce production of ROR YT and transcription factor that is the member of retinoic acid receptor family. Roles is the T cells restricted encoded by ROC genes. So sometimes the protein may be called ROCs. Inflammatory cytokines, notably interleukin 6, activate the transcription factor STAT, which function with ROR gamma T to drive the T helper 2 response. T helper 17 cells appear to be abundant in mucosal tissue, particularly in gastrointestinal tract, suggesting that the tissue environment influenced the generation of this subset, perhaps by providing high local concentration of T growth, transforming growth in factor beta and inflammatory cytokines. This observation suggests that T helper 17 cell may be especially important in combating intestinal infection and in the development of pathological intestinal inflammation. The development of T helper 17 cell in the gastrointestinal tract is dependent on the gut microbes. In mice, commensal bacteria related to the Clostridium species are present, are potent inducer of T helper 17 cells. Function of T helper 17 cells. The T helper 17 cells combat microbes by recruiting leukocytes, mainly neutrophil, to the site of infection. Phagocytosis by neutrophil is the major defense mechanism against many common bacteria and fungi that can survive outside cells but are killed in phagolysosomes of neutrophils. T helper 17 cells play an important role in defense against this infection by bringing neutrophil to microbes. Most of the function of T helper 17 cells in host defense are mediated by interleukin 17, but other cytokines produced by this subset may also contribute. Interleukin 17. Interleukin 17 is an unusual cytokine because neither it nor its receptor is homologous to any other known cytokine receptor pairs. The interleukin 17 family includes 60 structural related proteins, of which interleukin 17A, interleukin 17, and interleukin 17F are the most similar. And the immunological function of this cytokine family are mediated primarily by interleukin 17A, and interleukin 17A is produced by T helper 17 cells as well as innate lymphoid cells 3 and some of gamma delta and CD8 T cells. Interleukin 17 receptor are multimeric and expressed on wide range of cells. We have seen already in chapter 7. They activate the nuclear factor kappa beta and other transcription factors. So this is the development of T helper 17. We see interleukin 1 and interleukin 6 produced by antigen presenting cell, and transforming growth factor beta produced by virus cells activated as transcriptional factor ROR gamma T and STAT3, which stimulate the differentiation of naive T cell to T helper T helper 17 subset. Interleukin 23, which is also produced by antigen presenting cell, especially in response to fungus, stabilize the T helper 17 cell. A transforming growth factor beta may promote T helper 17 response indirectly by suppressing T helper 1 and T helper 2 cells, both of which inhibit T helper 17 differentiation not shown. Interleukin 1 produced by T helper 17 cell amplify this response. So we have seen this. We have the bacteria and fungus. Then they activate the dendritic cell. The dendritic cell will produce interleukin 6, interleukin 1, 23. These all they can act together in naive T cell, and they can activate role. And this role, when it activated, now the naive T cell is changed to T17, T helper 17 cells, of which this one can produce number of interleukins, the 17 and 22. Hopefully we'll see the function. So interleukin 17 is an important link between T cell mediated adaptive immunity and acute inflammatory response, which we discussed in chapter 4 as one of the major reactions of the innate immunity. When T helper 17 cells are activated, these reactions are more severe and prolonged than what is seen in innate immune when T cells are not involved. So we'll see this one, and the structure again, figure 10-12. This is the function of T helper 17 cell. Cytokine produced by T helper 17 cells stimulate local production of chemokine that recruit neutrophil and other leukocytes, increases production of antimicrobial peptide defensin, and promote epithelial barrier function. Antigen presenting cells and the colon stimulating factor also they are involved. So what happened here? We see that we have the bacteria or fungal, because that's the main thing that helps in activating the naive CD4 T cells to produce the T helper 17. Then this one acted to the antigen presenting cell acts to the naive T cell. The naive T cells then it proliferate, differentiate with everything then happening, we get the T helper 17 cells. The T helper 17 will produce interleukin 17 and interleukin 22, which both of them they will go to tissue cells, and in here they will recruit number of interleukins as well. There will be recruitment of chemokines, the leukocytes, and tissue cells. They will lead to the production of tumor necrosis factors, interleukin 1, interleukin 6, colon stimulating factor. They'll be there, and this one lead to the inflammation. The inflammation also recruits neutrophils. Neutrophils are very nice in fighting against bacteria and fungal. So tissue cells, antimicrobial peptide, they are produced, and increased barrier function also is stimulated. So interleukin 17 has several important functions in host defense. 1. Interleukin 17 induces neutrophil-rich inflammation. It stimulates the production of chemokines such as interleukin H and other cytokines such as tumor necrosis factor to recruit neutrophil and to a less extent monocyte to the site of T cell activation. It enhances neutrophil generation by increasing the production of granulocyte colon stimulating factor and expression of its receptor. Recruited neutrophil engage and destroy bacteria and fungi. Interleukin 17 also stimulates the production of antimicrobial substances including defensin and the numerous cell types. So other T helper 17 cytokines. Interleukin 22 is the member of type 2 cytokines family, produced by activated T cells, particularly T helper 17 cells, and by some natural killer cells and the innate lymphocyte cell. The interleukin 22 receptor is heterodimer, in which one of the chains is also a component of interleukin 10 receptor. It is signaled via JAK1, TYK2, and STAT. Interleukin 2 is produced by T helper 2. Interleukin 22 is produced by T helper 17 cell in epithelial tissue, especially for this of the skin and gastrointestinal tract, and serve to maintain epithelial integrity mainly by promoting the barrier function epithelial by stimulating repair reaction and by inducing production of antimicrobial peptide. Interleukin 22 also contribute to the inflammation, in part by stimulating the epithelial production of chemokines, and may therefore be involved in tissue injury in inflammatory diseases. Interleukin 21 produced by activated CD4 T cells, including T helper 17 cells and T follicular cells. It has a wide variety of effect on B and T cells and natural killer cell. Interleukin 21 receptor belong to the type 1 cytokine receptor family, consists of ligand binding chain and the YC gamma C subunit and activate JAK-STAT signaling pathway, in which STAT3 is especially prominent. An important function of interleukin 21 is in antibody response, especially the reaction that occur in the immune center. Interleukin 21 stimulates the generation of T follicular cells and activate B cells in the immune center. Some of the other reported action of interleukin 1 include increasing the proliferation, differentiation, and effector function of CD8 T cells and natural killer cell. Roles of T helper 17 in host defense. The principal function of T helper 17 cell is to destroy extracellular bacteria and fungi, mainly by inducing neutrophil inflammation. The recruited neutrophil ingest and kill extracellular microbes. The importance of this role of T helper 17 cell is illustrated by inherited disease called Job syndrome, a hyper IgE syndrome, which is caused by mutation in STAT3, resulting in defective H17 development, and is characterized by increased susceptibility to the cutaneous fungal and bacteria infection. Patients present with multiple bacteria and fungal abscess on the skin, resembling the biblical account of punishment visited on Job. Defective T helper 17 function is also associated with chronic mucocutaneous candidiasis. Patients with autoimmune disease, autoimmune polyendocrine syndrome produce autoantibody against their own interleukin 17 and also develop candidiasis, seen in chapter 15. Surprisingly, patients with mutation in the ROCK genes, which encodes ROL gamma T, the conical transcription factor for T helper 17 cells, show defect not only in interleukin 17 production but also in production of interferon gamma, the classical Th1 cytokines. The T helper 17 cell contribute to the pathogenesis of many inflammatory diseases. T helper 17 response has been associated with psoriasis, inflammatory bowel diseases, rheumatic arthritis, and multiple sclerosis. Agents that block the development or function of T helper 17 cell are very effective for the treatment of psoriasis and are being tested in other diseases. The antagonists are not beneficial in Crohn's diseases and inflammatory bowel diseases. So, despite abundance of T helper 17 cell in the intestine, the role of these cells in Crohn's disease and Crohn's disease is uncertain. In some chronic inflammatory diseases, both interleukin 17 and the interferon gamma contribute to the disease. These two cytokines may be produced by a subset of interleukins of the T helper 17 cell that are thought to be highly pathogenic, or by a mixture of T helper 17 T helper 1 cell in lesion. T helper 17 cell also helping to maintain integrity of epithelial barrier, such as in the intestinal tract. This function is partly because these T cells limit the entry of infectious microbes through the barrier by stimulating local production of antimicrobial peptide, and partly because interleukin 22 promotes the regeneration of epithelia. It is possible that different populations of T helper 17 cells are involved in this protective function and in pathological reaction caused by this subset. The function of other helper T cell subset. Although T helper 1, T helper 2, and T helper 17 subset are the best defined effector cells of the CD4 lineage, several other populations of cytokine-produced T cells have been described. Some of these are CD4 T cells, and others do not belong to either CD4 or the CD8 lineage. Other subset of CD4 effector cells. Other subset of CD4 plus T cells that produce various cytokines have been described, but their importance is less clear than the Th1, Th2, Th17 cells. Th9 cells Activation of CD4 T cells in the presence of cytokines, tumor growth, transforming growth factor beta, and interleukin-4 generate population of effector cells that produce mainly interleukin-9 and therefore called the TL+9 cells. The TL+9 cells are abundant in skin and mucosa tissue. Interleukin-9 activate mast cell and have many other reported activities. T helper 9 cells have been implicated in allergic diseases including atopic dermatitis and asthma, in defense against parasitic and numerous other reactions. T helper 22 cells. T helper 22 cells were defined as CD plus T cells that produce interleukin 22, known cytokine T helper 17, but not interleukin 17. The function of interleukin 22 were discussed earlier. The role of this subset in host defense or immunological disease are not established, in part because it has not been possible to selectively eliminate any of these subsets or their product. Furthermore, a unique transcriptional signature of these cells has not been well defined and possibly exists that they are transient state in differentiation of a more established subset. Other cytokine producing T cell. In addition to CD4 plus and CD8 T cells, there are a small population of T cells that have distinct features that may serve a specialized function in host defense. The best defined of these lymphocytes are gamma delta T cells and natural killer cells, natural killer T, NKT cells, and mucosa-associated invariant T cell, might. All three of these cells type have common characteristics that distinguish them from CD4 and CD8 T cells. They recognize a limited number but a wide variety of antigen, many of which are not peptide. And these antigen are not displayed by class 1 and class 2 MHC molecule on antigen-presenting cells. The antigen receptor of gamma delta T cells, natural killer T cells, and mucosa-associated invariant T cell have limited diversity, suggesting that all three cell types may have evolved to recognize small group of microbial antigen. It is also possible that these cells mainly respond not to particular antigen but to cytokine produced at sites of infection and tissue damage. Because of these features, these T cell populations are often said to be at the crossroad of innate and adaptive immunity. All three cell types are abundant in epithelial tissue, such as gastrointestinal tract. Their function may include the following. So they are involved in early defense against microbes encountered at the epithelium before adaptive immune response have developed. They're involved in surveillance against stressed cells, such as cells that have undergone DNA damage or are infected, and elimination of these cells. They are also involved in the production of cytokines that influence later adaptive immune response. Gamma delta T cell. The antigen receptor of MHC restricted CD4 and CD8 T lymphocyte is heterodimer composed of alpha and beta chain. There is a second type of clonally distributed receptor composed of heterodimer gamma and delta chain, which are homologous to the alpha and beta chains on T cell receptor found on CD4 and CD8 T cell lymphocytes. T cells expressing gamma and delta T cell receptor represent a lineage distinct from more numerous alpha beta expressing T cell. The percentage of gamma delta T cell vary widely in different tissue and species, but overall less than 5% of all T cells expresses this form of T cell receptor. The gamma delta heterodimer associated with the CD3 and the zeta protein in the same way as T cell receptor alpha beta heterodimer do. And the T cell receptor induced signaling event typically of alpha beta expressing T cells are also observed in gamma delta T cells. Although in theory the potential diversity of gamma delta T cell receptor is even greater than diversity of alpha beta T cell receptor, in reality only limited number of gamma and delta V region are expressed, and there is little or no junctional diversity. Different population of gamma delta T cell may develop at distinct times during ontogen, contain different V region in their antigen receptor, reside in different tissue, and have limited capacity to recirculate among these tissue. In mice, many skin gamma delta T cell develop in neonate life and express one particular T cell receptor with essential no variability in V region, whereas many of the gamma delta cell in vagina, uterus, and tongue appear later and express another T cell receptor with different V region. The limited diversity of gamma delta T cell in many tissues suggests that the antigen recognized by these receptors may be conserved among cell types or microbes that are commonly encountered in this tissue. One intruding features of gamma delta T cell is their abundance in epithelial tissue of certain species. For example, more than 50% of lymphocytes in the small bowel mucosa of mice and chicken, called interepithelial lymphocyte, are gamma delta cell. In mouse skin, many of the intraepidermal T cell expresses gamma delta receptor. Equivalent cell population are not as abundant in human. Only approximately 10% of human intestinal epithelial T cells expresses a gamma delta T cell receptor. Gamma delta T cell in lymphoid organ expresses more diverse of T cell receptor than in epithelial dendritic cell, in epithelial gamma delta cell. Gamma delta T cells do not recognize MHC associated dipeptide antigen and are not MHC restricted. Some gamma delta cells clones recognize small phosphorylated molecule, alkyl amine, or lipid that are commonly found in mycobacteria and other microbes, and they may be presented by non-classical class 1 MHC molecules. Other gamma delta T cells recognize protein or non-protein antigen that do not require processing or any particular type of antigen presented for their presentation. Many of gamma delta T cells are triggered by microbial heat shock protein. A working hypothesis for the specificity of gamma delta T cell is that they may recognize antigen that are frequently encountered in epithelial boundaries between the host and external environment. A number of biological activities have been ascribed to gamma delta T cells, including secretion of cytokines and killing infected cells. But the function of these cells and their contribution to the normal immune response remain poorly understood. It has been postulated that this subset of the T cells may provide a protection against microbes at the epithelium before the recruitment and activation of antigen-specific alpha-beta T cell. However, mice lacking gamma delta cells created by deletion of gamma or delta T cell genes have little or no immunodeficiency and only a modest increase in susceptibility to infection by some intracellular bacteria. These cells also may be involved in inflammatory disease. For instance, in inflammatory skin diseases, psoriasis, gamma delta cell appear to be among the earliest interleukin-17 producing cell in lesion. It is not known if this is the case in other inflammatory disorder or what gamma delta cells are recognizing and how much they are contributing to the development of the disease. Natural killer T cells. A small population of T cells called natural killer T cells expresses markers such as CD56 that are found in natural killer cell. The T cell receptor alpha chain expressed by a subset of natural killer cell have limited diversity, and in human these cells are characterized by a T cell receptor alpha chain with a V region that is encoded by a rearrangement of V alpha 24 JAR18 gene segment with little or no functional diversity associated with a T cell receptor beta chain that uses one of the three V beta gene segments. Because of this limited diversity, these cells are also called invariant. This is the invariant natural killer T cell. Invariant natural killer T cells. Other natural killer cells exist either that have a quite diverse antigen receptor. All natural killer T cells are the natural killer T cell receptor recognize a lipid that are bound to classical class 1 MHC-like molecule called CD1 molecule. Natural killer T cells and other lipid antigen-specific T cells are capable of rapidly producing cytokines such as interleukin-4 and interferon gamma after activation, and they may help in marginal zone cells to produce antibody against lipid antigens. Natural killer T cells may mediate protective innate immune response against some pathogens such as mycobacteria, which have lipid-rich cell walls. And invariant natural killer cells may even regulate adaptive immune response primarily by secreting cytokines. However, the role of these cells in protective immunity or disease are unclear. The mucosa-associated invariant T, the myeate cell. Myeate cells are another subset of T cell that expresses an invariant alpha-beta T cell receptor that uses a rearrangement of V alpha 7, 2 JAT3 gene segment. Myeate cells recognize fungal and bacteria metabolites of riboflavin synthesis pathway presented by non-polymorphic class 1 MHC-like molecule called MR1. MHC class 1 related protein 1. Most myeate cells are CD8 and can be activated either by MR1, this MR1, restricted presentation of microbial riboflavin derivatives, and directly by cytokine including interleukin 12 and interleukin 18. The effector function of myeate cells includes secretion of inflammatory cytokines such as interferon gamma and tumor necrosis factor and cytotoxic against infected cells. Myeate cells are found in blood and gastrointestinal tract and the liver. They account for about 50% of all T cell in human liver. Given their abundance in the liver, it is possible that they represent an important barrier to gut flora that have breached the intestinal epithelial barrier and entered the blood, because blood draining the gut first enters the liver through the portal circulation. Having concluded our discussion on the function of CD4 effector T cell and some less common T cell population, in Chapter 11 we will consider effector cells of CD8 lineage, whose major roles are in defense against viral infection. Summary. Cell-mediated immunity is adaptive immune response stimulated by microbes inside host cell. It is mediated by T lymphocytes and can be transferred from immunized to naive individual by T cell and not by antibodies. Naive CD4 T cell lymphocyte may differentiate into different types of specialized effector T cell, including T helper 1 cell that secrete interferon gamma, which mediate defense against intracellular microbes, T helper 2 cells that secrete interleukin 4 and interleukin 5, which favor the AGE and eosinophilus mast cell mediated immune reaction against helminths, or T helper 17, which promote inflammation and mediate defense against extracellular fungus and fungal and bacteria. The differentiation of naive CD4 T cells into subset of helper T cell is induced by cytokine produced by antigen presenting cells, by T cells themselves, and by other cells. The differentiation program is governed by transcription factor that promote cytokine gene expression in T cell and epigenetic changes in cytokine gene lock, which may be associated with a stable commitment to a particular subset. Each subset produces cytokine that increases its own development and inhibits development of other subsets, thus leading to the increased polarization of response. The T helper 1 cell recognize antigen of microbial that have been ingested by phagocytes and activate phagocytes to kill microbes. The activation of macrophage by T helper 1 cell is mediated by interferon gamma and CD40 ligand and 40 interaction. Activated macrophage kill phagocyte microbes ingested into phagolysomes by the action of reactive oxygen and nitrogen species and enzyme called classical macrophage activation. Activated macrophage also stimulate inflammation and can damage tissue. T helper 2 cell recognize antigen produced by helminth and other microbes, as well as environmental antigen associated with allergies. Interleukin 4 promote B cell isotope switching and production of IgE, which may cause mast cell degranulation and inflammation. Interleukin 5s secreted by activated T helper cell activate eosinophil to release granules content and destroy helminth, but may also damage host tissue. Interleukin 4 and interleukin 13 together provide protection at epithelial barrier and induce alternative form of macrophage activation that generate macrophages that control inflammation and mediate tissue repair and fibrosis. The T helper 17 cells stimulate neutrophil-rich inflammatory response that eradicate extracellular bacteria and fungi and may contain the integrity of epithelia. T helper 17 cell also may be important in mediating tissue damage in autoimmune disease. Alpha delta T cells, natural killer cells, and mucosa-associated invariant T cells are T cells that expresses receptor of limited diversity and recognize various antigen without a requirement for major histocompatibility complex associated presentation. These cells produce cytokine and may contribute to the host defense in inflammatory diseases. That marks the end of this discussion, and we will continue in other chapters. And below they are selected the readings that you can add up and increase your knowledge regarding this topic. So our next topic will be differentiation and function of CD8 plus effector T cells, and we will continue soon. Thank you. Enjoy. Jeremiah Puzo.